Hello friends, in this video we will discuss about the application of biotechnology. Now the first one application of biotechnology, this biotechnology is important for medicinal purpose or also biotechnology used in the agricultural purpose. Now this agricultural use of biotechnology is a very important role in agricultural crops. Now this one of the genetically modified organisms are produced through this biotechnology. Now the first one application is a genetically modified organism, or it is called the GMOs. Now these are some living organisms whose the genetic material has been artificially manipulated in a laboratory. Now these living organisms, some specific genetic material, they can also conserve or preserve artificially, or they can manipulate the specific techniques through the genetic engineering. Now some organisms, now the created combination of plants like some animals, plants, bacteria, these are some organisms or these living organisms or some viruses. They are not occur generally or naturally. Some gene are not expressed, or some traditionally some breeding methods, hybridization methods, cross breeding methods is also applying that times not produce the genetically modified plants. Now one of the techniques are using through this biotechnology, the one of the terms like the transgene or transgenic plant. Transgenic. This transgene or transgenic means the insert the gene or genetic sequences of plants. It is called the transgene. Now the GMO, genetically modified organisms or genetically modified plants are produces some aims of genetically modified organisms, some aims, important aims to introduce new traits. They introduce the new traits to the plants. The new traits or new characters are introduced to the plants. They are not produced naturally, artificially they can introduce some gene or some characters of plants. Now this plant is also produced the high quality or nutritional values or some resistant varieties or insect pest or disease resistant varieties are also produced. And now they can also produce the drought resistant plant, increase the high yielding capacity and improve the nutritional values. All these characters all in one plants. Now these all characters are showing in one plants, and it is the most important aim of the genetically modified organisms. Now the genetically modified organisms are like the transgenic plants. Now the first one terms transgenic plant. This transgenic plant. Now the transgenic plants means the desired gene is isolated, a particular gene or this desired DNA. This desired DNA is isolated from plants. This or the other organism desired particular gene, and this particular gene they can inserted to plant. This transgenic plant means one of the particular transgene or the in gene sequence of the DNA, they can desired gene of the DNA isolated, and this gene or the desired DNA they can introduce to the plant cell. Now this plant cell, this particular gene is inserted through the vectors or this plasmid, this one combination or this plasmid vector, this plasmid or through the vector, this particular gene or desired DNA they can introduce to the plant. Now through this plant cell, this particular gene is introduced. Now this is one of the techniques, or this techniques is also the transgenic plant is produces. Now the in human beings, now the human being rear is a very dependent on agriculture, and as world population is increases day by day, and demand of this plant, like the artificially producing or improved varieties of plants are produced through this biotechnology. And now the traditionally this biotechnology is used, but traditional breeding programs is also involve the sexual process. Now these breeding programs which result also the high quality of present day of plants like the wheat, rice plants, corn, potato. These plants traditionally some improved high quality foods are produced, but recently the biotechnological approach have been applied to these plants, and they can create the genetic characters. Now the first transgenic plants, the first transgenic plant is produced as tobacco. This is one of the transgenic plants. The first transgenic plant is produced. The first transgenic plant is tobacco. This tobacco plant is the first transgenic plant produced, and now the more 60 transgenic plants are also produced through this method, or genetically modified plants. Some 60 transgenic dicots plants, monocots plants like some wheat, maize, oat. These monocots are also some vegetables like the tomato, soybean, potato, sugar beets, grapes, some fruits like grapes, apple, cotton. Some are the transgenic plants is also produces. Now these transgenic plants have look of the bioreactors, and now for the molecular forming. Now this is the first one, it is transgenic plants. Now this first transgenic plant is tobacco, and also the various 16 plants also the transgenic. Now the some advantages of genetically modified plants. Some advantages of GMOs. Now the next point, advantages. Advantages of genetically modified plants. Now the genetically modified plants or food plants. Now the first one use or first one advantage of genetically modified or food plants. Now the first, insect pest resistance. The first advantage is insect pest resistant varieties are developed. Resistance, insect pest resistant. The one of the bacterium is important for this production of insect or pest resistant varieties. Now the beet crop. This beet means Bacillus thuringiensis. It is one of the gene beet. The Bacillus thuringiensis is one of the bacterium, and this bacterium, it is an aerobic type of bacterium, and this bacterium Bacillus thuringiensis, it is one of the gene have present the gene like a cry gene, present the cry genes. Now this beet gene or this Bacillus thuringiensis, it is one of the aerobic soil bacterium, and this aerobic soil bacterium it is a spore producing bacterium, and they can produce the several exo and endotoxins. They can produce the toxin. Now this bacterium has a particular gene, and this bacterium particular gene is called the beet gene or it is known as a CRY gene. This CRY gene is found in this bacterium. Now this bacterium, the proteins, the protein is synthesized. This protein is synthesized in the crystalline forms, and now these crystalline forms, this protein is present, and it is dissolved in alkaline solution or alkaline pH. This crystalline form of this gene is present, and now this gene, it is dissolved in alkaline pH, and this protein, then through the toxic, this protein become toxic. And now this CRY gene or this crystalline form of proteins, it is causes, they can cause the toxins. And now in plants, the pH is not alkaline, and that's why they can do not cause the plants. And now this gene, the beet gene, it is isolated and they can introduce the plants. This particular one of the genes, specific gene, it is isolated. The gene is isolated and introduced to plant through the transcription and translation process This one of the beet gene is isolated and introduced to the plants, and now this is one of the crystalline form of the proteins are found in this gene, and this crystalline gene, it is dissolved in the alkaline pH. Now they can introduce to plants, and this gene is found everywhere in the plants. Now sometimes the caterpillar larvae is bite on these plants, or this one of the pest caterpillar, that is a larval stage. This caterpillar is a larval stage and of the pest, and they can feed on this leaf of the cotton plants, and now this protein, they get into the guts of the pest, and now this pH is alkaline, and this present in the alkaline form in the guts of this paste, and now this alkaline pH, this CRY gene is converted into the toxins. This protein get converted, crystalline form of protein is converted into the toxins, and now the toxic effect, protein get toxic, and now the activated this toxin, they can bind to the surface of the mid guts of the epithelial cells, and they can create the pores, they can causes the cells, now the swelling or lysis of these cells. Now this gene or this one of the protein, they get inside the pest body, and that time they can enter through the digestive system of mid guts, and now this mid gut, the mid gut of digestive system, and some epithelial cells are found inside, and these epithelial cells form these are the epithelial cells, and this toxin or protein enters, they become toxin, and swelling takes place in these epithelial cells in the mid guts, and they can create the causes, and now this swelling or lysis of these cells, and that's why the result that become this paste is a die or the death of the paste. Now these beet toxin genes, they can introduce into the crop plants, and the beet toxin resistant varieties are also improved through this biotechnology. Now it is the most important advantage of agricultural biotechnology in insect pest resistant varieties are also developed. These insect resistant varieties like the beet toxin resistant to bollworm, and now this beet cotton or BT cottons is the first genetically modified crops of our countries is produces. Now the second application or second advantage in genetically modified crops, next one advantage is a nutritional, improve the nutritional qualities or biofortification. Improve nutritional quality, nutritional qualities, for it is called the biofortification. The second advantage in agriculture, improve the nutritional qualities of plants. Now the transgenic plants have been produced to provide the nutritional value. These transgenic plants introduce the gene, specific nutritional gene also introduced to plants and develop the nutritional value of plants. And now the millions of people are suffering for some diseases. Some diseases for the deficiency of vitamins, and now they are essential for some malnutrition is very common in our countries. And now the improve the nutritional value or nutritional quality of plants. Now the one of the disease blindness that result from the lack of the vitamin A. This vitamin A is abundant. The vitamin A deficiency causes several diseases. Now the vitamin A is produced abundant in the milks and the vegetables. For carrots, for example carrots. And to solve this problem, success for the researcher related to the transgenic plants or transgenic rice varieties are also developed. These transgenic varieties like the golden rice, it is one of the transgenic plants. This golden rice plant. These rice varieties are developed. And now this transgenic, another one of plants of transgenic is a mustard, the golden mustard, also golden mustard plant. These two, one is a rice variety, rice and mustard plant. These two plants are produced, the nutritional value is increases to these plants, and these are the two transgenic varieties, rice and the mustard, golden mustard varieties are improved. And now the vitamin A, this golden color is due to the vitamin A content, and now the reduces the diseases association with vitamin A deficiency. And this vitamin A also introduced to the improve the nutritional value of plants. For example, golden rice and golden mustard. These two plants improve the nutritional values. Now the another one, improve the oil contents. Another one important nutritional contents like the oil content, the oily qualities, the oily crops like the soybean, oil palms, sunflower has been achieved by the transfer the Arabidopsis gene in this plant, transfer the specific gene oil improvement or the improve the oil contents of these plants, oily plants. And now this is one of the adapter or transfer genes in these plants. Now the another one nutritional value is also increase iron deficiency. Now the iron deficiency is also the serious nutritional problems, and they can affect it, and estimated 30% of the world population for the production of this transgenic crop that will produce the food rich in irons. Now the iron containing storage proteins are produced. Now the one of the iron containing protein like the ferritin, this iron containing protein, ferritin, this is the iron contained protein. Now this iron deficiency is also another problems, and now the this deficiency is controlled through the one of the gene is produced, the ferritin. This is one of the gene. Iron containing genes are formed and this ferritin is found in the many animals, plants, also the bacteria. This gene is found, and now this ferritin is isolated, and now this gene, the ferritin proteins, they can isolated from the another plants like the soybean or phallaceous, and has been this gene they can transfer to the rice. Now this ferritin gene they can isolated from animals, bacteria or other plant. Some plants they can also found this gene, means iron containing the genes, and they contain of the iron, and now this genes is isolated from different plants, and now they are insert this particular gene in the this rice varieties, and this rice variety develop the nutritional value. Now in this video we shall learn about these genetically modified crops. Now the genetically modified crops they are important for development in the agricultural field. Now the agricultural field, some plants improve the nutritional values and good quality of the foods. Different insect pest resistant varieties are also develops. It is a two important advantages of the genetically modified organism.